Imagine lysosomes are primarily in these low-numbered object types, and mitochondria are primarily the high number. This is just a toy example. The idea is that if we have these fundamental distributions, and we're given some mixed pattern, that we can back out what the percentage of the protein was between the two patterns. Okay, so how do we learn these object types? It builds on work that was done by Ting Zhao years ago now, which is that we find all the objects in the fundamental patterns all together, describe each object by features that measure the object properties, size, shape, distance from the nucleus, and so on, cluster the objects, and then use AIC to find the right number of clusters to represent them. And therefore, that allows us to represent each fundamental pattern as some kind of count or probability of observing each object type. All right? So here are just examples of object types from a large collection of images, just to give you a feeling that the clustering is working properly to find these different types of objects. And now, then we'll talk about the heart of the issue for this particular paper, which is various mixing models that we can try to consider. So what we're going to say is that we've learned from example images of the various fundamental patterns a distribution of each of the object types. Now, this is a distribution in the form of numbers of objects. Okay? So 27 objects of type 1 and 33 of type 2 and so on. And that's this FP. And this will be averaged over many images of that particular fundamental type. And that then we have some mixing fraction that says how much of a given protein is contained in each of these fundamental types. And what we observe for that mixed pattern is an object distribution, which is just the sum of the product of those. Okay? So that what we have then is, if we the fundamental patterns originally were 100% small round for one pattern, 100% small long skinny for another pattern, if we mix those two together, we form a pattern that is some fraction of one and some fraction of the other. And of course, the sum of the fractions has to equal 1, and we can solve this problem in a very straightforward way. That should say constraint, with the constraint that all of the fractions are greater than 0, greater than or equal to 0. Now, an alternative is to consider that any given object distribution is drawn from a multinomial distribution, and then we can find this multinomial distribution just simply by counting how often each object type for pattern P of type I is observed relative to the total number of objects in pattern P. And then once again, the object type probabilities for a mixed pattern are just the sum of the object type distributions over all of the patterns. And we can find the values of these mixing fractions that maximize this criterion using Newton's method. There's a closed form solution. The third thing we can do, both of those models consider that the objects themselves are the units that we're working with, but in reality, actually, we might be interested in the fraction of fluorescence that's contained. So these methods would give us the fraction of objects of a given type, but in fact, we're interested in, more likely, the fraction of fluorescence, or the amount of protein, the mass of protein that's contained in each type. And we can do exactly the same thing, which is that we can build a vector that's a fraction of the pattern P is contained in each of the object types from 1 to k. So this is now the fraction of fluorescent in each object type. We once again have some mixing fractions, and we have a linear mixing model that says how much of the fluorescence we would observe. Not how many objects, but how much of the fluorescence we'd observe, and we can solve that in a straightforward way by pseudo inverse. Okay, now, here we come to the heart of things. Given this algorithm, how do we test whether or not this works? One of the ways we originally tested it was by making synthetic images, where we took known objects from known patterns and mixed them together in known proportions, and then tested to see whether it got the correct answer. The problem with that is that obviously is going to work because we're using the same mixing model to generate the test data as we use R for backing it out. What we need, though, to realist it is patterns where we know they're mixtures and we know what the mixture fractions are. And that's something that doesn't exist, because if we actually had data on what fraction of a protein was in these various compartments, we wouldn't need this method in the first place. That's what the method's for. So we created a test set by mixing different proportions of two fluorescent probes that have the same fluor, but that localize to different parts of the cell. And these are just example images of these two probes. Lysotracker stains acidic compartments like lysosomes. Mitotracker that stains mitochondria. And here's an example of an image of a mixture of the two dyes. Okay? So what we did using automated microscopy in collaboration with the colleagues at the Genomic Research Institute of the Novartis Foundation, generated a large collection of images for various mixtures of these two different probes, and made a grid of const one. So then from the images of the pure probes, we learned the object type distributions. We found 11 distinct object types. This is the distribution of the mitochondrial probe in black and of the lysosomal probe in white, showing that these object types are relatively enriched in mitochondria, these object types are relatively enriched in lysosomes, and this gives us the basis for saying we should be able to unmix any pattern by looking at the proportions of the different object types. All right? So here are the results of doing that. This shows the layout of the data. The mitochondrial probe decreases in concentration in this direction. The lysosomal probe decreases in concentration in this direction. So this is a 50-50 mix of each. This whole diagonal is equal proportions of the two probes. This side over here is entirely lysosomal probe, and this row down here is entirely mitochondrial probe. So those rows are the ones we use for training the system. And then we evaluate all the other wells. Each one of these boxes, by the way, is one well on a multi-well plate with that particular combination of probes. And here is the results of the three different methods I told you about. And you can see that the linear unmixing, and it's a little hard to tell on this, and the fluorescence fraction unmixing both work quite well at recovering the results. And here are representing that in a different form, which is what we're estimating as the amount of a particular probe that was added to a well versus what was actually added, and you can see that there's pretty good agreement between those. So this represents, for the first time, a system that, given an image that has a mixture of two different patterns in it, that it will estimate for you with reasonable accuracy what those percentages. Now, the model assumes that whatever images we use for training the model, the images of the pure patterns, that the images of any mixed patterns are indeed mixtures of those training patterns. But what if it's the case that, for example, there's now a third pattern or a fourth pattern that is contained in some image that we're trying to unmix? How do we check to make sure that we sort of reject that additional pattern? So what we do is two things. When we find each object in a test image, in a new image, we check whether or not that object is likely to have actually been of the type that it's closest to in the feature space. And if it's too dissimilar, then we say that object type isn't even among my library of object types. I'm going to consider that to be an outlier. And we add all those objects and their associated fluorescence into an unknown category. So that means that we can unmix. We can get, this is pattern 1, this is pattern 2, and this is all the stuff I don't know what it is. All right? And so this just shows you that using this type of test, if we take, this is the accuracy of unmixing the mito and lyso data that we just looked at. When we use a threshold on this distance from the object type, or the probability that this object is from that particular type, as we raise the threshold, what we will end up doing is eliminating real objects that are actually of that type to increasing degree, and we will therefore, for the real data, for the true lysosomal data, the accuracy of unmixing, the recovery will decline slightly. But here it shows for a third pattern, which is actually nuclei, that we can reject all of the nuclei, the nuclear pattern, as being rejected. So none of these nuclei are considered to be a member of the known patterns. Okay, so that worked reasonably well. The other thing we can do is if this new image contains significant amounts of that unknown pattern, then the error of the unmixing will be large. When we measure the reconstruction error, that error will be large. And so what we do, we measure that reconstruction error. We say, how well did my learned fractions multiply times the mixing, the fundamental patterns, give me back what I actually measured? And if that reconstruction error is above a threshold, then we consider the whole image to be unknown. All right? That we say we weren't able to adequately unmix it. And this shows testing of that, again, as we use a higher and higher or more and more stringent threshold on what we consider to be a good unmixing. And in this case, we tested it with images of a fourth pattern, that is an endoplasmic reticulum pattern. And we were able to reject the endoplasmic reticulum images with very high fraction rejected, while we were still able to accept 85% of the true images, the lysosomal images. Okay? So that tells us that we can do a good job of estimating these outlier patterns. So therefore, when we say that this is truly a mixture of this protein and this protein, or this pattern and this pattern, that it's correct. Now, very briefly, we tested this on a real high-content screening assay. This was an assay for effects of the drug baflomycin on autophagocytosis. This is a type of high-content screening assay that is typically done by classification. We did it by unmixing, and what you can see is you get a very good dose response. Those are the three differences. A very good dose response curve from measuring the fraction of protein that's in the autophagosomes and not in the presence of this drug. So this represents an approach for that. So to summarize, to begin summarizing, given images of these various fundamental patterns, and then given some image that we want to analyze, we can do one of two things. We can either use traditional classification approaches, this is the by now standard approach to do things, and say, this thing is closest to this one, therefore I'll call it Golgi. Or now, we can use the supervised unmixing approach and get out the fractions of the protein that are in these different places. And this is a much more informative way of describing the pattern. And if we look at, for example, in UniProt or any of the other protein databases, we may see Go terms that say a particular protein is present in Golgi, or maybe it's in Golgi and ER, but you won't find any kind of estimates of how much of the protein is in those various compartments. And this gives us, for the first time, a way of representing that. Okay, now, we are releasing today open source software that does this task, either as a MATLAB source or as compiled binaries. And it supports training on images either from your local disk or from Omero servers. For those of you who know, Omero is an open microscopy environment, public open source project. And so this software is available from our website. Just kind of give you a quick overview. The basic idea is that you load some set of images, you give it a name, and then after you've done that, for in this case 2 patterns, you give a name to the model, and it goes and you train the model. It shows you how much of the different object types the protein is in, and then it estimates the amounts of the fluorescence in the various compartments. Now, this was a significant advance, but in fact, one of the problems is that we may not know all of the fundamental patterns, or we may not have markers that are entirely in just one of them. And so the best approach to that is unsupervised unmixing, which would be to just take a collection of images and simultaneously learn the fundamental classes as well as the fractions. And Tao Peng will be giving a talk, a proceedings talk tomorrow on a system that he and Luis Cuello developed for doing that. Now, one last piece. We now can say, here are these various patterns. Here's how much of the protein is in each of these patterns, but how do we communicate the patterns themselves? As an aside, we've developed a system for building generative models that describe the pattern so that you can take images of a particular pattern, build a model of a cell in which you have a nuclear model, a cell membrane model, and then a protein object containing model that allows us to build synthetic images of each of these different proteins. And so what we propose as the sort of final deliverable with respect to all of this process, the answer to the question, how do we represent information about this problem, is that we have collections of images, we learn the fundamental classes, we learn mixing fractions, and then for each of the fundamental patterns, we learn a generative model that enables us to synthesize images that are drawn from that pattern, and the combination of those two gives us the ability to do cell simulations with accurate geographic descriptions of the proteins and accurate fractions of the protein between these various compartments. So I will stop there and call your attention for those of you interested in things like this. In Bioinformatics 2010 will be held in CMU in a couple of months. There's, of course, a session in the preceding track that Gene Myers and I put together also on this bioinformatics topic. I will also call any of the graduate students in the audience your attention to the Fellows Program, and I'll stop and thank you for your attention.
Could you comment on the of this
Pattern
number also
we are able to that has this many. In test experiments, we were able to unmix up to 8 different patterns with 83% accuracy. So it scales reasonably well to a number of different patterns, and it's absolutely scalable to as many proteins as you want. And, the only issue is in the supervised case, having examples of all the different patterns that you want to run. But we absolutely believe it is in fact scalable, and we're applying it to proteome scale collections of images.